From the indifferent gonad, testis differentiates under the influence of the SRY gene that works as a transcription factor to other genes. Under a coelom epithelium, which will become the epithelium of the peritoneal membrane, we got the tunica albuginea as the connective tissue layer that will become gradually thicker and thicker. But more importantly, we got a proliferating medullary cords of the former coelomic epithelium. They become independent, and they contain the gonocytes that have found their way into the primordium of the gonad. And they are surrounded by the mesodermal epithelium. So first, these are solid epithelial cords with no lumen. And they are moving into like inside of the testis. That's why they are called medullary cords. Moreover, from the mesenchyma here, the Leydig cells will differentiate either individual cells or frequently in small groups. Otherwise, we got mesenchyma here, mesenchyma tissue. So let's label the proliferating medullary epithelial cords. This would be the mesoderm of the dorsal body wall cavity, the coelomic mesoderm that will become the peritoneal membrane. This is the tunica, the connective tissue tunica albuginea, which contains mostly collagen, bundles of collagen fibers. And these proliferating medullary cords contain the gonocytes. They will differentiate in testis into spermatogonia. Moreover, we got Leydig cells that differentiate from the mesenchyma here. These proliferating medullary epithelial cords will differentiate into seminiferous tubules of testis, and also into the rete testis. So only to remind you, the seminiferous tubules of testis, they contain, in the basal compartment, they contain the spermatogonia, like stem cells for the spermatogenesis that starts with the onset of puberty. And these cells develop into the supporting Sertoli cells that are touching each other forming the testicular barrier, and these Sertoli cells will, for example, support the spermatogenesis once it starts. And in the interstitial space, we got the Leydig cells with lots of smooth endoplasmic reticulum and lipid droplets, cholesterol droplets. This is typical for steroid-producing cells because the smooth endoplasmic reticulum, did I say rough? No, smooth endoplasmic reticulum. That's where the formation of androgens and other steroid molecules occurs, and the cholesterol is a precursor of these steroid hormones. So let's label the structures. This will be spermatogonia, this will be the supporting Sertoli cells. The supporting Sertoli cells are producing the androgen binding protein, abbreviated as ABP, that locally increases the availability of androgens, and also they produce the anti-Müllerian substance, or Müllerian inhibiting substance, that results in the suppression or regression of the paramesonephric duct. When the testis develops. Moreover, I should note that the lumen here appears in the puberty. Right? These are seminiferous tubules, and the lumen here appears with the onset of the puberty, together with the spermatogenesis that will happen here later on. But now we're talking about embryonic testis. We should also mention the Leydig cells. The interstitial Leydig cells that are producing androgens since 8th week, and these androgens will contribute to the growth of the mesonephric duct, and the mesonephros, and other organs of the male phenotype. Without the effect of the androgens, most of the organs would differentiate into the female phenotype, because there would be the effect of the maternal and placenta estrogens. So it's necessary to have these androgens to develop the male phenotype correctly. Let's have a look on the proportions between the testis and epididymis. At first, well, the epididymis originates from the mesonephros, from the lower pole, which actually at first is larger than the testis. If this will be the testis with its seminiferous tubules that are convoluted, and then they are straight, and then they meet in the mediastinum of the testis, forming the labyrinth of the rete testis. Then the mesonephros, which is a larger organ with its mesonephric ductules that are drained by the mesonephric duct, because it serves as an excretory organ first. Now the upper pole of the mesonephros will regress and disappear, but here the mesonephric tubules will join the rete testis. They will connect. They will find each other. There are some chemoattractants, like factors, that will help to establish connection between these tubules. So this is testis. This is the region of the rete testis. And this is the mesonephros. And the mesonephric tubules, many of these. They will actually join the rete testis. This is the mesonephric duct. And we should not forget the paramesonephric duct, which will be somewhere here. Yeah, the Müllerian duct. Now the paramesonephric duct, under the influence of the Müllerian inhibiting substance, it undergoes regression. So I will also cross the duct, the paramesonephric. With the further development, the testis will become larger with its lobules, seminiferous tubules. The rete testis, as we know, is now connected to the former embryonic mesonephric tubules, but now we call them efferent ductules of testis. And we know that they are arranged in the head of the epididymis. And they gradually can join into a single duct, which is the former mesonephric duct, but the single duct is many times folded here in the tail of the epididymis, and then it leaves as the deferent duct, or ductus deferens. So this is the testis. Sometimes small residuals of the paramesonephric duct might persist as the so-called appendix testis. It does not have any apparent function known to us. Yeah. So there might be residuals in the form of appendix testis, but more importantly, here are the efferent ductules of testis. So the mesonephric tubules will become the efferent ductules of testis that are arranged in the head of the epididymis. They gradually join the former mesonephric duct, which becomes the duct of epididymis. But it's extensively folded, so you still can see it as on multiple sections or profiles on a histological section. And this would be the ductus deferens. That will later on become part of the spermatic cord. So the mesonephric duct forms the duct of epididymis, the ductus deferens, and also seminal vesicles and the ejaculatory duct. Epididymis is therefore the surviving lower part of the embryonic mesonephros. Now the testis doesn't stay in its anatomical position where it originated in the abdominal cavity, but it goes down. We call it the descent of the testis. Briefly, before the descent, we got the umbilical cord here. Here's the genital tubercle. Here would be the abdominal cavity. And somewhere here, up, there will be the embryonic testis, which is connected to the dorsal body wall by a structure called the genital ligament. And that continues as a connective tissue stripe called the gubernaculum of testis. Here will be the pubic symphysis, and here will be the rectum, with the anus and the perineum. So this is the genital tubercle. This will be the symphysis, pubic symphysis. This will be the anus. This is the testis, attached by the genital ligament. And the connective tissue cord called gubernaculum. The gubernaculum does not grow, so during the further grow, relatively the testis is pulled into this region. Well, the genital tubercle enlarges because it will form the penis, and also here in this region we got lateral scrotal swellings that will become the scrotum. Here will be perineum and the anus with the rectum. And also the peritoneal membrane forms a vaginal process here in this direction. Here will be the pubic symphysis. And the gubernaculum extends to the scrotal swellings. So the testis is somewhere half its way to the scrotum. It will then go through the inguinal canal. So if this is abdominal cavity, this will be the vaginal process. Of the abdominal cavity. This is the scrotal swellings. And this is the testis, and the gubernaculum is a fibrous tissue cord. And finally, the genital tubercle becomes the penis. The scrotal swellings become the scrotum. Here will be the perineum with the anus. The communication between the abdominal cavity and this vaginal process will be closed, obliterated, but there will be a small serous cavity of the scrotum surrounding the testis and the epididymis that will be here as well. Yeah. So this will be the penis, the obliterated communication, closed communication. The testis went through the inguinal canal into the scrotum, into the scrotal sac, where is the testis, and there is the serous cavity of the scrotum. With two layers, with the parietal and the visceral layers. Now, the presence of the testis in the scrotum is routinely checked when a newborn male newborn infant is born. The rest of the gubernaculum is known as the scrotal ligament. Sorry, the spelling here is scrotal ligament. All the other coverings of testis are also derived from the abdominal wall, including the skeletal muscle and all the fascia, etc. So the presence of the testis, as I said, is checked, and in 97 of full-term newborns, it's there. If not, it needs to be definitely found where it is, whether in the inguinal canal or did it remain stuck in the abdominal cavity or somewhere, because it could be a problem.